The Conversational Flow, a KCAS podcast.
Hello, and welcome to the 20th episode of The Conversational Flow, an official podcast of KCAS Bioanalytical and Biomarker Services. KCAS is a bioanalytical CRO serving the pharmaceutical and biopharmaceutical industries for over 40 years. Unlike KCAS' other podcasts, the Weekly Bioanalysis, which features news and topics more generally associated with the world of bioanalysis, the Conversational Flow focuses more on the niche of flow cytometry and its role within the greater industry. My name is Adam, and I've been in the flow cytometry space for over 15 years. I'm here with my co-host, Brian.
Thanks, Adam. Brian Weil, General Manager at KCAS Bio, and I've been in the flow cytometry CRO industry for about 13 years.
I'm wondering, Brian, if we should update that because we've been doing this for over a year.
We have. I was thinking about that. Like, if we're at episode 20, it means we're almost at 2 years. So yeah, we better go back and review.
Brian and I are members of the growing leadership and scientific advisory team at KCAS. Either or both of us are available to answer any questions you may have regarding this podcast or any of KCAS services. While our KCAS headquarters are located in Kansas, with additional locations in Europe and Australia, we're both here in Philadelphia, and Jeremy, our producer, is in Missouri. So we're truly all over the map. We're elated to have you listening to the 20th episode of our podcast, which will be available virtually everywhere. Wherever you choose to find and play your podcast, you'll find the Conversational Flow. We welcome all of you, whether you're joining us for the first time or if you're a regular listener to any of our other podcasts. Today's episode will be a review of the latest news and resources, and then a focus on a topic of our choosing, before discussing any feedback we receive from you. We're constantly looking for topics, and we'd be happy to discuss something that you want us to cover. So again, we're fired up to have you here, and we're looking forward to a fun episode today. To kick us off for episode 20, Brian is going to go over news and resources before diving into today's topic. Brian?
Thanks, Adam. For this time, we came up with a collaborative agenda here.
Yeah, I do want to make sure we're going to have to unpack agenda this time, right? Because the last time we let everybody know that I did the agenda, which is different than normal. And I'm not taking any bonus points here for doing so. But what I did want to cover is your comment that the last podcast was terrible. And so why don't you tell us about that?
So last podcast had multiple things in it that then manifested themselves as truth, right? And that's my main objection to the agenda from last time. So we talked about, I think, the shingles vaccine or some sort of vaccine, and then this past month I did get a case of shingles, which was just terrible, just the worst.
Yeah, it is. And I sympathize. As we talked about, I had it before. But man, you got it extra bad, if that's the thing.
That's it. Just on the face. Again, it's a very good thing that we're doing this as a podcast, recording only, face for radio sort of thing. So, yeah. So between that and then also, I feel like as soon as we said that there's been no big autoimmune acquisitions, there was a big autoimmune acquisition. So, yeah, I mean, between the shingles thing and then, us taunting the mergers and acquisition gods, I just had some issues with the last agenda, but it's all right.
So what you're saying is this agenda, we should talk about how we still have not won the lottery.
Oh, I didn't even think about using that for our own benefit, but you're right. Yes, we should have. All right, next time.
Yeah. See, so self-serving. Anyways, please continue with the agenda.
Yes, indeed. So today we've got some medical news from AACR and the FDA, some investment news, again, from the FDA, but also from Sanofi. And then, of course, some KCAS news with some upcoming meetings and a pretty cool paper. So in terms of jumping right in, AACR is the American Association of Cancer Researchers, and it's basically the premier cancer meeting for the year. And that's got a mix of drug developers, of course, CROs, but then also a lot of clinicians, right? And that's where they're learning about what is best practice with treating cancer and, some of the main readouts from previous years from new and interesting drugs.
Yeah, and so they had a really interesting one with Keytruda. Obviously, Keytruda's been out for a long time. Pembro, as it's also known, or pembrolizumab, which is a humanized antibody. It's a PD-1 inhibitor, and so prevents binding of its ligand PDL1 to PD-1. And what they were talking about was the trial where it was demonstrated that for stage 3, stage 4A resected, locally advanced head and neck squamous cell carcinoma, that there was significant positive results there. And this is the first time seeing positive results in this sort of indication for over 20 years.
And that's just a huge point to make, right? Like, the fact that this indication hasn't really seen improvement. So that means, first of all, that our current standard of care is doing pretty good if it's hard to beat, right? But then also that all of this work that's being put into R&D and innovation, things like that, hasn't translated for changes in patient care.
Yeah.
So having this that is that real change helps point to the effectiveness of these drug development efforts.
Yeah, it really does. And I was interested too in learning a little bit more about PD-1, Keytruda, and how all that works. Specifically, what they talked about in this study was that they had the assessment of expression level of PDL1 in these cancers for the various patients. And they did see somewhat of a stratification of PDL1 positive versus PDL1 negative, or at least below 1% positive PDL1, and showed that if there was a higher level of PDL1 expression, that there was better benefit. Now, this is similar to other indications where Keytruda is being used. I found interesting, though, that they did say some PDL1 negative tumors do still respond to Keytruda. It's always something that, as a scientist, you're curious about how that works, what the mechanism of action is there.
Right. Like, are there just more active T cells, or is it just triggering a different component of the immune system to do something, or is it helping to get through like the cold zone around a tumor and just enhancing activity in the correct place? As you say, like the mechanism there is always interesting.
Yeah, yeah.
Um, yeah, but certainly like the ability to use this as a way to prevent new cancers as well. So, you do this, and then you also add in either radiotherapy or chemotherapy, and then you just see significantly improved survival, event-free survival as well. That was the main point of this article. So, just an excellent outcome there. And of course, the flow cytometry angle is that this is all based on triggering the immune system, using the immune system to help fight cancers. And the more we understand that, the better we understand that, the more we can target these types of therapies.
And one of the things they talked about too here was treating with Keytruda prior, then removal of the tumor, then continuing treatment with Keytruda. They had, plus or minus the chemotherapy, as you mentioned. And so they were trying to parse out then which angle, which part of that therapy was most effective or most necessary, whether having it beforehand did seem like that was very effective, and then having it afterwards as well.
Yeah. No, exciting times. And it really just shows as well the number of different degrees of freedom with these treatment regimes, right? Like, you can treat with this drug, how much of this drug, exactly when, how many times. And then on top of that, like, can you use additional drugs in combination with this one? Maybe with Keytruda, it's great, but then maybe in combo with another drug, we could see even better improvement.
Yeah, and interesting too, they talked about the percent increase or improvement here, and now at one point mentioned about 11%, but that's significant, right? Where some of the therapies that have been approved demonstrated 2 to 3%, which still you're talking about more people being helped, and so every tick forward there is a big one.
Absolutely. Yeah, so exciting news and hopefully more to come and more studies around how Keytruda can help more people and or similar therapies as well. All right. And then, in terms of the next one, I guess on the back end of that, the question is how do we translate that into care? And one part of that process is always an FDA review. So the second story we've got here is just an FDA news release around the first AI assisted scientific review.
Yeah, this was cool in and of itself that the FDA is piloting AI use, essentially deploying it with full integration set by the end of June, excuse me, to have a secure generative AI system that's integrated with the FDA's internal data platforms. And what they're trying to do is remove or reduce the amount of busy work that scientists have to free them up for more decision making items. Now, I will admit that I kind of went down a rabbit hole as I usually do with stuff like this, and they were talking about generative AI, and like, I want to understand more about the different AI. So here, generative AI has models like large language and generative adversarial networks (GAN for short) that are trained on large data sets with the goal to generate relative output that makes sense or relevant output that makes sense. Now, I heard something like a generative adversarial network, and I'm like, that sounds really cool. So I looked into it. It's got two neural networks, and one of them is the generator, one is the discriminator. And the way it's set up is like practically a game where the generator adds fake data into the real data, and then the discriminator needs to determine the real from fake. And it goes back and forth like that to be able to continue to evolve the understanding, the information there. And I'm not like a AI guru or anything like that, so this is kind of the surface level understanding here. So apologies if I'm not getting it exactly right, but I think that is the broad stroke. And yeah, it was really cool.
Very interesting, right? And as the press release itself says, like the promise of AI has been out there for a long time. We've known it's coming. We've known that we need to be applying it in different contexts. So it's interesting to see the rubber finally meeting the road.
Hmm. Yeah, and this is everywhere now, right? Frankly, if you're not working on some way to embrace AI, you're frankly falling behind. The efficiencies that you can gain are very real. Obviously, you've got to be careful with it. You've got to test it, make sure that it fits your needs. You can only deploy it in certain areas, right? But when you do it responsibly, you can save a lot of time and really push things forward faster.
That's it. It's really interesting, and I wonder what it was like when accountants first started rolling out Microsoft Excel, right? Like,
Mhm.
was it the same thing where everybody was like super excited about it and thought you could use it in all these different places, and then they did, or was there pushback against it and fears that it would mess up the calculations or something else?
Come on, everything gets a pushback, but I do remember the days of being in a lab with graph paper and putting my first job out of college in an academic lab at the Albert Einstein College of Medicine. And like I'm sitting there graphing stuff by hand. And now we've got Excel. I could put all that stuff in, the amount of work that I could do now. But that's the thing, too. It comes with an increased expectation, right? You've got to turn over a whole lot more work
Always
because you
does.
got
Always
these
does. Yep.
tools.
Yep. They say we spent all this money on this system. Now do like 3 times as much work, right? So yeah. All right. But, exciting times. And hopefully this will help the FDA be super efficient, get approvals through more quickly. And, as we know, like we've been applying it in a couple of places at KCAS or evaluating its application in a couple of places at KCAS. So definitely, as you say, it's reshaping the industry. And basically, if you're not at least experimenting with it at this point, then you're behind the curve.
So are we delinquent though in getting through a whole segment on the FDA employing AI and efficiency without having a single joke about government and all of that?
Uh, no, I think we're okay. And the next segment is also going to be about the FDA. So we've got ample opportunity. There's time. We'll pace ourselves.
There you go.
Yeah. So and this now goes into investment news, right? And I think we would be remiss if we didn't at least mention some of the churn that is impacting the biotech sphere at this point. So
So were you just like really dying for lots more articles that you had to do 3 in this one item here?
that's correct. I thought to myself, I am not doing enough work. Adam's summarizing his whole journal article, and I'm only doing like 4 tiny little sections on the website. So I'm going to do more in this one. So I made a mega thread, and part of that was, as you say, just to add more content. But part of it too was that I couldn't find a good summary of exactly what this means because a lot of it's too new, right? So that does make it challenging.
Well, that's the thing. Nobody really knows. There's a lot that needs to be unpackaged here. Right now, all the layoffs. What does that mean for the FDA? One of the articles you have here is talking about how things are going to be moved to Europe or other countries because of the layoffs slowing down things at the FDA. But as we just got through, the FDA is using AI, so hopefully that means they're more efficient. And
Yeah, and maybe just to introduce the section. So this first investment news is just the U.S. regulatory megathread, is what I'm calling it, and just with the tagline that it's a challenging time to be a biotech CEO, right?
mhm.
Um, and I do want to be relatively neutral as we approach it because anytime a new thing happens, it is difficult to assess the full impact.
Yeah.
And that's why I say it's very challenging because I'm sure all of these CEOs need to go and tell their investor what this means. And CEO and investors are never okay with the thought of we need to wait and see, right? They want to know what to do with their money now. So as a CEO, you have to have an answer for some of these things. And, you know, as a podcaster, we can be relatively lax about it and say, like, these are all changes and there's a lot of things going on. But it does definitely make it a very difficult investment environment because you don't know exactly how some of these things are going to be shaking out. So, as you said, there are 3 articles that I included in this section. There were a number of other ones that we could have as well. But for this one, we included one on the changes at the FDA where a lot of people have changed roles or left the agency as point one, and what that can mean.
Mhm.
Point two is the tariffs that were added and then modified and things like that. And then point three is the most recent with the executive order to attempt to rein in drug costs.
Right. So each one has differing impacts, but what we're trying to do here, as you alluded to at the outset, is look at it, take a step back and look at it from a more global perspective. What will this do to the market? We're not trying to read the future or anything like that, any tea leaves or tarot cards or whatnot. I'm not really any good at that kind of stuff, but I think so. Your first one, the FDA upheaval pushes some biotech firms to plan early trials outside the U.S. That's where I was alluding to that. Some of these smaller companies may just not have the ability to do their early trials in the US. They may have to go to other places. It's too early, really, to say. So consultants are mentioning that they're seeing some interest in going this direction, but then the discussion around the EMA essentially saying that right now they're not seeing much of a shift just yet, but it's too early to tell. Um, you know,
That's
uh,
it.
Go
Yeah,
ahead.
I mean, between that, I mean, with all these things, if you think about the time it takes to plan a trial or plan a drug launch, um, even just on the trial, which is a little bit easier to think about, usually there's between 3 to 12 months of development time before a trial is launched. As you plan out, where are you going to get the patients? How are you going to get the drug to them? What tubes are you going to collect? Is the clinical trial protocol approved? All the different I's and T's that you need to dot and cross before you can even dose a patient. That's a long process, and it's not something that can change on a dime. So any of these things that happened in between now and January, really we haven't seen the impact of as of yet. So all these are leading indicators, right?
Yeah, and I mean your point's well taken. Cost is one driver of where you're going to have your clinical trial. There are many others, and so they could have an impact. They might not. Just kind of waiting to see. I am kind of curious. The last one that you have in there for the drug prices mentioned slashing them up to 90%. Uh, Specifically, there, how is that going to work since the executive orders don't override federal law? So there's probably going to be a good deal of fighting to be able to get to a point where there's an agreement on both sides. And so that's certainly another one that's going to have a long wait time before it's decided, I would expect.
I would expect as well, right? Yeah, and there's a lot of questions about exactly how some of these things are going to be implemented. The end result, though, of the whole is that the total price that you can charge for a drug may be limited, and the cost of producing a drug may be increased. And the combination of those two things makes investors unhappy. If you couple that with uncertainty or risk, then that is a difficult recipe that certainly will not help the industry here. Um, so, yeah, I mean, I guess to sum up, there's a lot of things to be concerned about or to keep an eye on, but it's unclear exactly how much of an impact they will have. So I guess we'll stay tuned, and hopefully we're giving a readout in December saying everything is a-okay. That would be the best best possible result there.
It is interesting, though, talking about drug prices, right? The US pays the highest prices in the world for prescription drugs, at least according to this article. And so the goal is to try to identify a most favorable nation policy or put in place a most favorable nation, which essentially means that the prices for the US will match other economically advanced nations, what they pay. So really curious to see how that shakes out. Always talk about healthcare in the U.S. and everybody's trying their best, but a system that certainly has room for improvement.
Certainly, yeah. I mean, if there's a way to drive down prices, then as a consumer of healthcare, that is great, right?
Well, and not only drive down, but while maintaining the expansive innovation that this country has had over the course of its time, right? Obviously a lot of these drugs are getting developed and made in the US, and you don't want to drive that innovation out of the country.
No, certainly not, right? And for all of our medical news stories, those don't exist unless there's the investment news story. So definitely we need both pieces to be able to function here as a healthcare system.
Mm-hmm.
um Yeah, and then outside of regulatory, just to follow up on our comments about the last agenda. Now, as soon as we had that podcast, then Sanofi immediately afterwards penned this deal for additional autoimmune markers or therapeutics here.
So what you're saying is Sanofi was listening to our podcast and they're like, well, I'm going to make these guys liars.
That's the only thing that I can think of that makes sense. That is it.
Yeah, right. So basically, they had what is it? Let's see, a deal with Erenadel Labs. Apologies if I'm butchering that there, but it's an AI research biotech, and they have two bispecific antibodies. So HXN1002. Now this targets integrin alpha 4 beta 7, so that's crucial for homing to the intestinal mucosa, and TL1 alpha, which is abnormally expressed in autoimmune disease. And so what the aim here is to treat ulcerative colitis and Crohn's disease. And then the other one is HXN1003, targeting again TL1 alpha and then IL-23, looking at colitis again, and then skin inflammation. Now, the thing that I dove into on this was bispecific antibodies. I know we talked about this before, and I will give a shout out to Arendelle Labs. They look at optimizing protein structures, and so really kind of using AI at that, pushing the envelope. We've talked about this before with what was it?
Recursion or
Recursion, yeah. We've talked
Mhm.
about this several times, right? And so there have been 12 bispecific antibodies approved by the FDA as of August of last year. That was the most recent data I'd found, of which 9 of them are the CD3 T cell engagers. And so these would be pretty unique among that subset, assuming, they continue to progress there.
That's it. And it's a nice intersection of everything that we want, right? There's AI, there's flow, there's anti-inflammatory, there's bispecific antibodies. So yeah, exciting times and really maximizing how quickly we can exploit the benefits of these bispecifics, things like ADCs. Once we know that something has a decent safety profile, then we can start to tweak it and figure out how it can attack different things. And that then aligns perfectly with Sanofi's corporate strategy of saying, like, we want everything related to immunology in their shop, which makes sense to me.
Yeah.
So
Yeah, and I was amazed too that the bulk of the bispecifics that are out there are produced by essentially 3 companies. There are a couple of others in there, but they were the ones that made up the bulk, and it just a really interesting technology. Each company has their own unique platform and how they work, how they just very cool.
yeah.
So, what do you got next?
Yeah, next up is KCAS news. So our KCAS news for this week is that you and Dimitri are going to CYTO,
Wait, this is news to me. I'm glad that we're
right?
putting this in here. No.
By the way, book that flight.
Book that flight. Yeah. So Dimitri and I will be at CYTO along with several other of our colleagues at KCAS. We'll be presenting on what is it? Phagocytosis oxidative burst assay. And then also too, you've got some news here on something called MAFCA.
MAFCA, indeed. So this is the Mid-Atlantic Flow Cytometry Association. So it's a new group that's forming up to ideally talk about flow in the Philadelphia area. And yeah, they'll have an event on the 12th of June, and a couple people from KCAS will make sure to be there because it's going to be right downtown.
Yeah, that sounds like fun. It's always good to have a group like that in our own backyard. I know there are other similar organizations a little bit further away for us, but getting like-minded flow cytometry people in a room and see what shakes out.
That's it. Yeah. So a lot of these conferences where people can get together and figure out what's next for the field. I always like quoting Ed Bastian, the CEO of Delta, on his thoughts on industry collaborations. Right. His big mantra was that the airline industry as a whole should never compete on safety. So everybody should make sure that airplanes are safe, that air travel is always considered the safest mode of travel, and that the whole industry and all manufacturers sort of agree on that as a central tenet. And then, of course, the different players are going to compete on price, are going to compete on the way they deliver their product and things like that. But nobody competes on safety. Like, that's not an acceptable parameter to say we are safer than Spirit or we are safer than a different like airline, because that drags down the entire industry and makes people think that it's a problem. So I like to think about that for flow cytometry and think, what does the entire industry need to agree that we don't compete on? And then what can we compete on again? And how do we, of course, from a KCAS perspective, show that we are good in those areas?
That's an interesting thought. I hadn't heard about that before, but it certainly makes sense, right? If one company is knocking the other's safety, what do you put in their mind? Oh my goodness! Like the consumer's mind is, wait, I didn't know it was that unsafe. Regardless of if we're talking about a minor chance, the thing I always hear is that. You're safer for skydiving. Let's put it that way. You're safer skydiving than you are on the trip to the place to skydive, right? But anyway, total tangent on that. So getting back to your point about flow cytometry, what does that mean for us? What should we not be arguing about? And what are your thoughts? I mean, where would you go with that?
My thoughts are that we need the equivalent of ICH M10, right?
Hmm.
Where that was just a pivotal moment for the ligand binding assay group, where they agreed that these are the parameters that you need to have in a validation report. This is what the report needs to look like, and this is how we're going to make them machine readable and translatable across studies. And in a very, very short time frame, it became the industry standard and a requirement for doing any type of LBA regulated work. So, to me, the wider adoption of things like the H62 guidelines or the further specification of those guidelines in a flow cytometry context would make the most sense.
Yeah, standardization, making sure that everybody is doing as close to the same thing as possible, because you don't want to have data that you can't trust. And how does that? We talked about soil cap before and things of that nature. I think that allows us to extrapolate information from unrelated studies that much easier, and forward information, forward knowledge.
That's exactly it, right? Like, how do we use the results of the Merck Keytruda study alongside the results of a Sanofi new collaboration on autoimmune cells? Like, how do we combine those things? Right now, there's no effective way to do that with flow cytometry, but you can imagine a future where, with appropriate standardization, more and more of these results can be compared across studies, definitely across drugs, potentially across indications, to help understand the immune system and how it reacts to disease and drugs both.
And so what you were doing here was just kind of keying off our paper for the month, huh? The standardization procedure for flow cytometry data harmonization and prospective multicenter studies.
It's like it's all related somehow, Adam.
How did you know that that was the paper that we were going to do?
Yeah, I mean, due to careful and thoughtful planning and foresight, right? So,
Yeah,
yeah,
so this is a paper in the Frontiers in Immunology. The impact factor is 3.8 as of 2023. And I mean, the title states it really, looking at ways to harmonize a prospective multicenter study. Now, it is interesting that we're talking about harmonizing 11 different flow cytometers. And we're not talking about like what we do, where we've got two SciTechs at our site and two SciTechs in the Lyon site and two SciTechs in Australia. And so it's all the same machine. We're talking about different machines. And that's just a entirely different beast. So tell me your thoughts there.
yeah, and you got to look at it in context, right? Like, the two big pieces for me are what level of precision is required, and B, what level of complexity is being assessed. So the level of precision for a clinical trial needs to be quite high, right? Like, if you want to make specific statistical inferences from things, then it needs to be, absolutely identical. So I can see the benefit of additional expense for easier to combine data in that sense. And then the other thing is complexity. If you're running a relatively straightforward panel, then it should be easier to harmonize across different portions there. Um, Whereas if we're running a very customized, indication specific flow panel for a very complex readout, that might be much more difficult in this type of cross center application with multiple machines.
And I do want to take a step back. When you say that it's got to be exact, we're not talking that one cytometer and another cytometer get the exact same number, exact same value for percentage or MFI, for the exact same sample. That's not the way this works, right? We look at it as percent CV, so coefficient of variation. And what this paper was setting out and trying to do was get a percent CV of less than 5%. Now, obviously, it changes across different markers. And so your percent CV would be higher for more rare populations. But I just wanted to clarify, we're talking exact. There is that realm of what does exact mean, and so there's a ballpark, right?
That's correct. Maybe I should say statistically exact. Maybe that's a better phrase, right?
Fair. Yeah. And so they did do a lot of testing here, but I think the important part is to understand the layers that they went through to be able to do this for all these different machines here. Starting out with the Versacomp capture beads and making sure that those look similar across the instruments, and then doing an intra-center normalization. So then again, more beads, the 8-peak beads, right? To look for changes within the instruments in the center, and then going through and here doing automated work, right? Automated gating after they do manual compensation, and all of that additional work, Python scripts to be able to then normalize between the sites. But on top of that, they also have processes that were there to support. They're using the Beckman Coulter Duraclone. So these are panels that are dried down. So they have a long shelf life. They only had to go through a couple of lots during the entire duration of the study, which helps, right? Because then you don't have to do as many bridges between reagents there.
That's it. Yeah, and you think about the amount of work that went into maintaining this harmonization across the course of the study, and it does become significant, right? Like you look at the cost of running all of the beads, you look at the cost of some of the reagents, and all of a sudden running the sample may only have been maybe 30 to 50% of the total cost of the work that went in here.
Yeah, we're talking 2,559 patients that were run during the course of 4 years, so 2014 to 2018. Yeah, that's a lot, to your point. And this goes back to what we were talking about before, the cost of clinical studies. It's not insignificant. And so if you're talking about things that either one are going to increase the cost or going to delay, any delay in getting a clinical trial started costs money, right? Because that is delaying that drug getting to market. And, it sounds insensitive to say it costs money when we're talking about patients' lives and everything like that. But also, it's a delay in getting that treatment to the patients, right? So it's a combination of factors there. And you really want to make sure that you're getting it done quickly. But the amount that it already costs is something that not a lot of companies can handle without sometimes getting investors or partnering with larger pharmaceutical companies.
Sure. Yeah. And so you see why this isn't usually done at large hospital centers, right? Because, do they need to harmonize with all these other things, or do they need to have a reference range and tell you where you fall on that reference range? And that's, I guess, the more common paradigm in clinical centers, whereas this is more of a standard clinical trial paradigm where we are doing things like quantitative bead assessments, inter-batch normalization, inter-site qualification, inter-instrument qualification on a regular basis here. So definitely applaud this group for starting to harmonize this data more and thinking about why they would want to harmonize the data as well from a clinical perspective, and the ability to then spin up these results into a larger useful data set.
And not only that, and they're not just comparing percentages, which is one level of harmonization that has its own intricacies, but then going further and being able to compare MFIs was another step. So it demonstrates just the amount of work that went into being able to accomplish something like this.
Yeah, that's absolutely right. All right. Yeah. Anything else that we need to mention about the paper? Did we go through the impact factor?
Yes, we did. 3.8.
Good.
No, I don't think that, I know it's kind of a broad overview. We link it so that if people are interested in the details of everything that went in, I mean, frankly, the figure one here of the paper and just going through each step of how they were able to harmonize is really impressive and helpful, I think, for people to be able to set up something like this. But really well done.
Yeah, indeed. All right. Well, does that bring us over to the next section of the podcast, or anything else that you wanted to tag on about harmonization and multi-center testing here?
Just that, we try to make it as simple on ourselves as possible when we harmonize across sites. It's more controlled for us to do that because the sites are under our supervision, right? So we get to guide how the processes are handled. The instrumentation is the same, the reagents are the same. And so we really go a long way in making sure that that data is going to be the same, to the point before, across each one of the sites as we receive samples, whether it's in Australia, whether it's in France, whether it's in the US.
That's it. And, even with all those advantages that you just mentioned, right? Like we own the sites, like everybody's using the same SOP, everybody's using the same reagents, everybody's using the same instruments. It's still like a lot of work. If I thought to myself, let's take like 19 different institutions, all with different SOPs, different people, different instruments, and see if we can get the same data. I don't know.
How many years do you think you would want to set that up?
It's daunting, right?
It is. It is.
Yeah,
It's a lot. That's why, really a lot of respect for this paper.
yeah, and for the consortium that backed it. So it is a European study, and this group was set up specifically to tackle exactly this type of question. So maybe that makes it easier if they were all set up, I guess, with that purpose in mind. So this is the Precise Ads. I hope I'm pronouncing that right, but Precise Ads Flow Cytometry Study Group. So apparently, this is basically a bunch of different institutions that are all sort of tied in and sort of voluntarily following the same procedures there.
Right.
So yeah, interesting stuff. All right. Well, I guess moving on, but then still keeping in with our overall theme of AI and standardization, things like that. Fan feedback request is: what is your favorite scientific AI system?
That's a good one. How many different ones do you use on the regular?
On the regular, I would say it is abnormal for me to interact with any less than 4 AI systems throughout the course of a day.
Oh wow, that's pretty good.
Right?
I have one that I prefer, but I will move into others depending on what it is that I'm doing. I've noticed that they tend to have strengths in certain areas.
Absolutely. Which one is your favorite? And we're biasing our fans here, but it's still a very interesting question.
I use ChatGPT most often. I've found that Claude AI has some strengths in other areas. I do appreciate that, and so I'll bounce back and forth between the two. I know our members of our team prefer Grok. I haven't used that yet, but certainly on the list there. What about you?
So, in terms of the one I use most frequently, I would agree that it's likely ChatGPT. But I will say, if I need wording, and specifically flow cytometry wording, I found Claude AI to be weirdly good at writing flow cytometry jokes.
Jokes. That's not exactly what I thought you were going to go
It's
for.
a random application of AI technology. Probably not what the FDA is using it for at this time.
No.
I'll send Marty an email and see if that's what he's doing. But yeah, I found that it is extremely good at exactly that.
I
Yeah.
I think it's hilarious that so your humor is coming from AI. Is that what you're saying?
I mean, I feel like the last 4 or 5 flow cytometry jokes have been at least AI influenced at this point. Like it is extremely good at using the word population in different contexts and making so it really seems like it understands, which is again very strange based on how AI works. But there it is. So again, brave new world.
Well, I mean, that's one of those things too that you could kind of get a comfort with after a while. So if you're engaging in it conversationally. And my family busts on me all the time because I don't know how many conversations I get out of without mentioning AI and the different things that I do with AI. And so it's well deserved. But I think you can almost get lost in the fact that you're talking to an AI instead of a person at times. Now, there certainly, I will say, when you're talking with an AI, there are things that you get feedback. You should challenge it, right? One of the best filters that I've actually been using is telling it no BS or no sugarcoating, because I'll ask it for feedback on something, and I hate when it gives me feedback that's all flowery. I'm like, oh, this is so great. I'm like, would you stop? I am not a baby. I want you to tell me the real deal here. Give me the real facts, right? And I've noticed that AI certainly sugarcoats a lot of stuff. So keep that in mind.
Fair. Duly noted. Yeah, hopefully some of the other AI solutions that we are implementing will also be sufficiently truthful there, right? So, and the FDA for that matter. Yeah, definitely.
Yeah.
Oh, goodness. But all right. So looking forward to some interesting fan feedback there as well. In terms of just outside of the scientific realm. Of course, I have a vacation coming up. This is the first time I'll ever go on a cruise, so we'll see about that. The word thalassophobia comes up. Have you heard of this fear before?
Oh, I'm so disappointed. I have, and I'm blanking on it. So I'm gonna have to let you spill it. And
The persistent and intense fear of deep bodies of water.
Yes, yes. So you have that?
I don't know if I have a persistent and intense fear, but certainly being over the ocean, just knowing there's like hundreds of feet or meters, or I don't even know how far, but it's a long way down. There's a lot of stuff in between me and the bottom, which is concerning to me.
Some places it could be miles.
That's it, right?
Now I will share a story then, because it's like the perfect segue for that. But when I was younger, I watched Jaws. Now, anybody who has watched Jaws should know or realizes that Jaws is PG, right? This,
Really?
yeah, I mean, that's one thing that's always been something I've wondered about. How in the world Jaws is PG? And so I was pretty young when I saw it. I mean, granted, I believe it came out right around I was born. So um, just take it for what it is. I think I watched it first when I was about 8, and my father would go to rent videos. And he's like, "What do you want me to get?" And I say Jaws, and just got tired of it at one point. And he said, they got other things aside from Jaws. I was like, "Do they got Jaws too?" He's like, "Forget it. I'm just gonna pick out my own." So, anyways, he takes us out on a boat. We're just like in a bay doing some flounder fishing, and he and my uncle and my siblings are, "Oh yeah, it'd be a great time to just hop out and go for a swim." I'm not going. My siblings jumped in the water. My dad's like, "Oh, you gotta go, you gotta go." So they tossed me in the water. And I swear to you, I did not go in the water. I bounced off the water back into the boat. Like that's just how much I was like, I am not going in that water. I watched Jaws. I know what happens.
Because that there's a shark coming straight up right now out of the water, and you just can't see it because it's so far down there. So I mean,
why was it only coming from me? My siblings survived; they're still here today. So I mean, thankfully, I was glad they didn't get eaten or anything like that. But all kidding aside, so you're going on a cruise ship, and this is your first time. So where are you going?
That's it. So it will go towards the Bahamas. So ideally, like not that deep of water. It's not like I'm going over the Mariana Trench or anything like that, right? But it is like the farthest I've ever been from shore, which again is mildly concerning, right? So we will see there. But you're right. Between Jaws and Old Yeller, I think that accounts for like 50% of childhood trauma, and those movies were not adequately marked for the amount of terror they've induced in the world.
It's funny because I just saw a cartoon about the book Watership Down and how it got translated into a cartoon. And because it was just a cartoon, parents brought this was like decades ago. And I read the book. It's a fantastic book, but really is very kind of bloody, right? There's a lot of messaging behind the rabbits in the story, and parents brought their kids to see the cartoon, and it was really bloody, right? It was a very good interpretation of the book. And so it just one of the, talking about scarring and damage, right? I'm sure there's a lot of kids that to this day are scarred from that. Not saying that it was bad in any way, the cartoon itself, but the book was fantastic. My mother put me onto that. And getting back to your cruise ship thing, I've been on a cruise once, went to Alaska, and it was only a week. I'm pretty sure I gained 5 pounds because the food there is fantastic, and take advantage of the excursions that they've got. Really, it was a lot of fun.
Okay, so you're saying don't be worried about the sharks down below because the ship is big enough that likely the sharks are not a concern.
I'm not promising you that, but
okay. All right, fair. It's realistic. I appreciate that. So, all right. What about you, Adam? Any other sort of deep-rooted fears like that?
No, I think that's, my primary one. I've gotten over it. I go to the beach, and there are times though my daughter pushes, she just starts swimming out further and further. And I said, listen, I'm sorry. There is a point where I am just not swimming out to get you. So please don't make me have to get that consideration going. Like I want to be able to save you if needed. So, yeah.
Fair. All right. Well, excellent. Wonderful episode as always. Thank you to all of our listeners. Our next recording is indeed episode 21.
Yeah, the big one. Our podcast can drink, as we said the last time, and I swear I don't drink as much as it sounds like I do on the podcast. But big 21, really exciting. I can't believe we do this once a month, and here we are, 21 in. So I get it. That tells you how many months we've done this for. But it's still cool.
Yeah, indeed. So thank you again for listening, and we'll tune in next week or month, whatever.
Thanks, everyone.
Yep. Okay. Bye.